So what is bio 3D printing? 3D printing is a process that varies, but material is ultimately just joined together by a computer in order to create three-dimensional structures. This tactic involves kind of a tedious process, but it can also be explained somewhat simply. So for this process, in order to recreate material, the printer needs something to copy. So some medical printers will isolate an extremely large number of human cells, for example. These cells can then continue and go through cell proliferation. These cells can then be loaded into a 3D printer and are ready to be copied. The printer then creates a pattern called a cell landing scaffold. The scaffold is then applied to many different beneficial applications like transplantation, drug discovery, and even disease study. Bioprinting in the sense of cardiac tissue engineering involves engineered blood vessels, heart valves, and also vascular networks. So to go even further in this printing process, biomedical engineers have to first successfully master and create a MATLAB code and digitally send this code to a computer that is linked to a 3D printer. The code then automatically generates what these engineers call a G code. The printer in turn receives the code controlling the printing nozzle, depositing the material into a desired structure and location. Some of the unique methods that were used in this article included a four-step process. So in step one, it kind of involved just the basic 3D printing process, but just went a step further in encapsulating endothelial cells. Step two resulted in the formation of the lumen-like structure in the vascular bed. Continuing on with step three, the researchers moved on with the process, but seeding the artificial material with cardiomyocytes, also known as cardiac muscle cells. And finally, with step four, the formation of a rapid population of vessel lining cells. So starting off with this first set of results, it seems a bit difficult to kind of comprehend at first, but essentially both data tables show the beating of the artificial or printed cardiac muscle tissue. The two tables also use the von Willebrand factor, so the VWF, which in turn is just really a large multi-domain that helps regulate the bleeding and blood clotting balance in the human body. The cells during the treatment also experience different dosages of doxorubicin, which is really just a bacterial antibiotic. So continuing on with the second set of results, the H data table shows a qualification of the CX43 expression, also known as the electrical charges, in order to impulse conditions with a change in electrical coupling. The cardiomyocytes are then compared on the four types of scaffolds, then plotted as percentages of area coverage. The second data table, so table I, shows a qualification of the angle of distribution of cardiac muscles on the four types of bioprinted microfibers. Overall, I thought the results aligned quite well with the data that was reported in the article. The data, like in graphs C and D, I think showed a pretty good representation of how the cardiac tissue and muscles were able to adapt and perform regularly over time, even after the procedure was completed. Data also showed the absolute value on the order of 20 to 30 kPa for the physical properties of the microfiber scaffolds, um, along with other materials throughout the research. So, in conclusion, I actually really enjoyed reading and researching more about this new and upcoming technology in the medical field. I have actually personally worked with the regular, just standard 3D printer, so I kind of had a small insight on what this article would be about, but I never really would have guessed all the power that this new piece of technology has for the world. So I think that this new ability to bioprint material will definitely benefit medicine in the future and will also help improve the life of others. Overall, a really great research opportunity and a great experience.